An important concept within the developmental origins of health and disease is what has been called the critical windows of sensitivity. This is also an important aspect when looking at developmental plasticity. And it has to do with the fact that not all periods during development are equal. Some times are more sensitive than others. And I'm going to try to give you some examples of this. But it's important for you to realize that during development there are lots of things that are happening and they are not all symmetric. That's what the concept of, it's called heterochrony, that different things happen at different times. And we'll see some examples of that. Obviously, if you affect a particular process at the time this process is actually happening at large, then your impact can be larger than if you do it at another period of time. One example that you are well aware of is what's called, it's the so-called fetal alcohol syndrome, right? The idea, the concept that mothers drinking alcohol during pregnancy can have long-term neurological consequences on the offspring, on the babies. But it's also equally well known that not all periods in pregnancy are equally sensitive. Some periods are more sensitive than others. And what's interesting to know is that the most sensitive period in which alcohol can be detrimental, can affect fetal development, is the first trimester. The first trimester, when many mothers do not even know that they are pregnant. That does not mean that a pregnant mother can drink later on. But the sensitivity to alcohol is much higher early in development, because that's the time that the fetus, or that the brain of the fetus is actually developing. The impact for neurological consequences is larger during the first trimester than later on. But we'll see some other examples. One example here has to do with the development of the penis in males. We know very well that the penis develops between week 7 and week 12. And we also know, or there's been reports of the use of progesterone to the mothers using progesterone. This progesterone can cross the placenta and alter the position of the urethral opening only in that time frame. This is what's called hypospadias. Hypospadias is a process in which the opening of the urethra actually is not placed on the tip of the penis, but along the penis. And this is a rather severe problem that can be surgically corrected, but it's an important problem that has been associated, among other things, with the administration of progesterone. And then you say, well, why would a mother take progesterone? Well, it's very typical, particularly in processes of in vitro fertilization, in which the corpus luteum is not actually providing enough progesterone during early development, before the placenta takes over of the production of progesterone. And in these conditions, it's rather typical that mothers receive extra progesterone. And that, of course, happens at around this time window. And I have looked at this, and I have not found any real consensus. Early on, there were some studies that suggested that this was the case. I have looked up more recent literature, and people is finding confusing results. To what extent, basically, progesterone actually is affecting hypospadias in babies. Also, of course, because we are now aware that this is a potential problem, there are ways and discussions on how much progesterone should be administered, if necessary, but I mean, you don't have to overdo it. So I think that part of the problem in the latest studies, the most modern studies on this problem, they don't coincide with the old problems, or with the old problem of hypospadias in the first studies that were done. But certainly, this basically suggests that something as the urethral opening, the opening of the urethra, the point where basically urine is actually excreted, can be changed, can be affected by different processes during weeks 7 to week 12 of gestation, and it can be affected by progesterone. the important thing here is the timing. Timing is the key. Within other time windows, it may not have any effect, but within that time window, it has. In terms of heart development, what you have here is the heart of a chicken at the hamburger Hamilton stages 17, 21, 25. And what you have here is basically an indication of different things happening to the heart. The one that I want to point out here basically has to do with this star that you see here, you see here, you see here, and you see here. And this is the development of the septum, the wall between the right and the left ventricle. Septation is actually completed here at the stage 33. So this is close to septation. What you see at this point is basically less of a septum. The septum starts and it's increasing in size and magnitude. This is one of the many things that are actually happening to the heart as the heart is developing. But there are many others. Ventricular trabeculation, the development of the epicardium. And notice that all of them have a window. They occur between certain stages. So different processes that can be affecting different stages can have a large impact on one of these specific morphological alterations in the heart. Third example, this is a classical example as well, has to do with the administration of Toledomind. Toledomind was a drug generated by a German company that was used as an anxiolytic and to alleviate morning sickness in pregnant women. So in 1957, it was rather extensively used in some countries, not all, in Germany, even in France. But for example, I'm not sure about France, but it was not used in Spain. It was prohibited in the United States. It actually never made it. So, but what happened here is that Toledomind actually binds and inactivates a protein called cereblon. And this protein has many functions, but one of them is actually the development of the limbs, the development of the fingers, the arms, the legs. If given at a particular time of development, Toledomind triggered or affected the development of the limbs, what has been called phocomelia. And this is basically a tennis player actually displaying the typical signs of phocomelia. Short arms, very underdeveloped. In this case, it was the administration of this drug that actually caused that. If Toledomind was applied or was taken by the mothers at different days post-conception, what they noticed is that the extent of this phocomelia, the extent of the problems was very different. Early on, it affected mostly the thumbs. Basically, the fact that there were fewer thumbs or the development of the thumbs. But as time went by, basically you could have phocomelia of the arms, phocomelia of the legs. So notice that within different windows, days post-conception, the use of Toledomind could actually have different effects. And that's because you were given a drug that was blocking a protein when this protein was most needed for the development of the limbs. Notice that if this drug had been given outside of these windows, perhaps the effect would have been much less. But of course, morning sickness in pregnant women is more acute the first months. And that's basically when it had the largest effect. This was a big scandal for the pharmaceutical company, etc. But from our point of view, the learning message here has to do with targeting a very specific window of development. With this drug could have severe permanent alterations because there was no way to catch up. Once this was affected, you basically cut off, destroyed the developmental process of the limb, and then it would not even continue later on because there was nothing to do. I mean, basically, the formation of the different bones, the structures that should be actually placed in that particular limb just did not take place. And that was the end of it. The last example that I want to give, and with this we will finish this lecture, has to do with memory formation, and this is a chicken example. It's a beautiful example, I believe, in which researchers have shown that if you give hypoxia to 14% for a whole day, for 24 hours, actually application of hypoxia certain days of development, but not others, impairs memory formation. And of course, this, for example, we know that developmental day 10 coincides with the beginning of axonal outgrowth, dendritic expansion, and synapse formation. So it would seem that day 10 is an important period for memory formation. Day 14 also coincides with the appearance of astrocytes. So what these researchers did is take chicken eggs, expose them to hypoxia for 24 hours, and then put them back in the normal incubator, hatch these chicks, and then at 2 days of age, or almost the day after hatching, they did memory tests. And what this memory test consisted of was a training period in which the chicken was trained to associate a red beet, a red glass beet, that had been dipped in some bitter substance as something to be avoided. And then they had the blue beet that actually tasted better. And then the animals, of course, had to perform selection, and then they chose which they selected. This was a sort of memory because the animals learned what to do, how to deal with, and then 2 hours afterwards, they trained them again. What they realized is that animals that have been exposed to hypoxia at certain periods during development could not remember what they learned 2 hours earlier. And then they picked the wrong beets, the bitter beets. This is what the data shows. And notice that this is actually rather large differences. This is the mean discrimination ratio after 2 hours of the training. So they actually, if the controls had a very good discrimination, they remembered very well which were the beets to actually choose. More than 90% did the right thing. But notice that if you exposed animals at day 10 to hypoxia, notice that the discrimination ratio drops to 55%. These animals here, the animals that were exposed to hypoxia on day 10, show evidence of a lack of memory. They just basically don't remember what they learned. The same thing at E14. What is interesting is that E10 and E14, as I said, coincides with some important periods for developmental neuronal growth that can be associated with memory. So I think that this is a beautiful example, again, of another critical window for developing something like memory. Hypoxia, we could summarize that hypoxia at times of intense synapse formation or neural specification have long-term effects on memory formation. Well, perhaps I should explain that, of course, what happens with hypoxia is that hypoxia is less oxygen, right? Less oxygen means that you are going to halt, you're going to stop some processes because there is not going to be enough possibility for actually, metabolically, there's not going to be enough substrate, ATP, to actually allow these processes to take place. So I think that this is the last example I wanted to give about the importance of critical windows. And this is something that, of course, when we think about the developmental origins of health and disease, we also have to think about which is the sensitive period for the process that you are interested in studying. And that's the end. Thank you very much.